How to develop a monoclonal antibody with step-by-step with an examples. Let's get start. Developing a monoclonal antibody, mAb, involves several steps, including the following. 1. Identification of the antigen. The first step is to identify the antigen, which is the target of the antibody. This can be a protein, carbohydrate, or lipid, among others. 2. Immunization. The next step is to immunize an animal, typically a mouse or a rat, with the antigen. This will stimulate the animal's immune system to produce antibodies against the antigen. 3. Isolation of immune cells. The immune cells, called B cells, that produce the antibodies are then isolated from the animal's spleen or lymph nodes. 4. Fusion of B cells with immortal cells. The isolated B cells are then fused with immortal cells, such as myeloma cells, to produce hybridoma cells. These hybridoma cells can continuously produce the same antibody. 5. Screening of hybridoma cells. The hybridoma cells are then screened to identify those that produce the desired antibody. This can be done using various methods, such as enzyme-linked immunosorbent assay, ELISA, or radioimmunoassay, RIA. 6. Expansion and purification of the antibody. The selected hybridoma cells are then expanded and the antibody produced by the cells is purified. 7. Characterization of the antibody. The purified antibody is then characterized to confirm its specificity, affinity, and other properties. Example. Suppose a scientist wants to develop a mAb against a specific protein found in cancer cells. The first step would be to identify the antigen, which is the protein in this case. The scientist would then immunize a mouse with the protein, isolate the B cells, and fuse them with immortal cells to produce hybridoma cells. The hybridoma cells would then be screened to identify those that produce the desired antibody. The purified antibody would then be characterized to confirm its properties and specificity. Note, this is a general overview of the steps involved in developing a monoclonal antibody, and the specific details and techniques may vary depending on the antigen and application.